So one of the things that we work on a lot in the lab is making hydrogels. And we use peptide cross-linkers and adhesive peptides within our gels so that the cells can remodel the hydrogel matrix. And so we actually make all of our peptides using this instrument here. It's very fancy. And so the way it works is that each one of these bottles holds an amino acid in an organic solvent, and you can tell the instrument which ones to put into the reaction vessel to build your peptide. And then once the peptide backbone is synthesized, we take the peptide out of this resin and we can modify it with whatever chemical moieties that we need in order to make it bind to our hydrogel.
My name is Eric Donders, and I'm an upper year PhD student in GRD Lab Tour. So my project is focused on drug delivery. Specifically looking at the final stage of delivery where the drug is at the cell that it's supposed to act on and is entering the cell so it can interact with its target protein. So in order to study this better and how we can improve drug delivery, I've taken a drug and made different chemical modifications to it so that it has different properties but hopefully the same drug activity. And so now what I'm doing is using some genetically modified cells that basically gives a readout for how the drug is entering the cells. I'm testing all of these different drugs to see how well they work. So this project has been really fun because I came into this lab not knowing any biology at all and having really limited chemistry experience with a chemical engineering undergrad. But through this, I've been able to do some really interesting chemical synthesis, and also I've learned so much about biology. So overall, it's been a really great experience, and yeah, couldn't have been happier with my decision to join this lab.
And I'm a second-year master's student in the GRD Lab. And my project is working on developing a methylcellulose hydrogel for the controlled release of a therapeutic protein to treat the ischemic brain tissue. And so today, I am dialyzing my methylcellulose modified with my weak binding peptide that will be used to control the release of my protein in order to remove any excess weak binding peptide from my solution.
All right, hey, so my name's Tim. I'm a second year PhD student here in GRD Lab. My project is on formulating polymeric nanoparticles for controlled protein release. And so what we have going on here is I've actually made a polymer last night. I'm going to purify into diethyl ether right now. And these polymers are going to be used to self-assemble to nanoparticles so that we can control release from a hydrogel. And so right now we're just in the synthesis stage. I'm going to be purifying my reaction mixture into diethyl ether. The reason why we do that is just so we can get rid of any unreacted monomers and just some other stuff I don't want from the reaction. So, right now we just got some cold diethyl ether. I had it on ice earlier. And here's my crude reaction. We're just gonna take this and we'll just slowly pipette it into our diethyl ether. I don't know if you can see or not, but this is pretty viscous and that's normal for most polymer solutions. So as you can see right now, that's all precipitating out. And yeah, as you can see, it's a bit of a slow process, but it's probably the easiest and the best way to do something like this.